Hi, everyone. I'm Crystal Van Hoof. I'm the Managing Director of Healthy Brains, Healthy Lives, and I hope that you have enjoyed the training day put on by Highball, Healthy Brains, Healthy Lives, and the Big Brain. So this is one of the two last sessions of the day, so hopefully you have some energy left because I think it's going to be a good one. For those of you who don't know, and I think, if you didn't hear Alan's talk this morning, Healthy Brains, Healthy Lives is a time-limited project that will end in mid-2026. And as we sort of come towards the end, we often have conversations about what the legacy of HBHL will be. And one thing that we all agree on is that one of the most important legacies will be the computational neuroscientists that we have been able to help to recruit to McGill, thanks to our startup funding. And you're about to hear from two of the startup funding recipients that we have from HBHL. So we have Blake Richards and Pouya Bashivan are going to talk to us about their research, and I'm very glad that they agreed to come to Iceland to share their work with you, which exists at the intersection of AI and neuroscience naturally. So Blake is an associate professor in the School of Computer Science and at the Montreal Neurological Institute, and is a core faculty member at MILA. And Pouya Bashivan is an assistant professor in the Department of Physiology at McGill. And so I'll turn it over to Blake. Thank you.
Thanks very much, Crystal. Hi, everyone. It's very nice to be here. It wasn't actually a hard sell to come to Iceland, of all places. I've been wanting to see Iceland for a long time, and of course, it's not too hard a sell to come here to speak to this group. So today, Pouya and I have been asked to talk to you a little bit about the use of artificial neural networks in neuroscience research. Obviously, artificial intelligence broadly is a very useful domain for application to the analysis of neuroscience data. And I'm sure many of you here use tools from machine learning for your research and in order to analyze data from the big brain or from other neuroscience data sets. But what we're going to talk about today is specifically the use of artificial neural networks as a tool in computational modeling. Now, I'm going to start us off by addressing what I think is one of the most important uses of artificial neural networks in computational neuroscience, and that is as a tool to answer teleological questions. Now, Jane informed me that I was basically playing with fire here by putting such a word in my title, so let me unpack this for you a little bit. So the word teleological, if you're unfamiliar with it, is a way of referring to things that ask about the purpose of a phenomena. My understanding is that the etymology of the word, just if you're curious, ties back to religion, when it referred specifically to questions that asked about why God put things together in the way that he did. But here, of course, we're not asking why God put together things in the way that he did, but rather perhaps we might be asking simply, why does a particular phenomenon exist in the brain? And so one of the ways of understanding that kind of question is to some extent, it's a question about the evolution of the brain. Why would natural selection have selected for creatures that have brains that do this kind of thing or that exhibit this kind of phenomenon? And obviously, teleological questions of this nature are very appealing. As scientists, I think we'd all like to know the why. You know, it would be nice to be able to say, you know, if someone asks, well, why does your visual cortex have separate dorsal and ventral streams? Why does the entorhinal cortex contain grid cells? Why do you have X, Y, Z in the brain? Whatever it is. And have an answer ready. Well, that helps us to do this. It would have been evolved for because it allows animals to do X, etc. But even though it's very nice to, we all, I think, desire the ability to answer these questions, it can be very challenging within neuroscience to answer them. And the reason that it can be challenging is because really we have no way of experimentally controlling the relevant variables for answering such questions. So, for example, let's say that I postulate that grid cells serve the purpose of helping animals to engage in vector navigation. That is, as a result of grid cells, I say animals can go from point A to point B without simply learning a series of stimulus associations, but instead actually having an internal map where they can calculate the best path to get from point A to point B. That's a hypothesis that people have put out about the purpose of grid cells. But how could you test that experimentally? In theory, what you'd need to do is somehow modulate or silence the activity of grid cells and only grid cells without affecting the other cells in the brain and demonstrate that animals lose this capability to engage in vector navigation. But that's not something you can experimentally do. Even with the most advanced types of optogenetics, you're not going to be able to accomplish that. At best, what you're going to be able to do is engage in some kind of correlational studies where you show, okay, like, you know, when these things are messed up, these other behaviors are messed up, etc. But really, it seems in part that it's almost impossible to experimentally answer such questions in neuroscience. But this is where artificial neural networks come in, and they provide a solution. What they do is they provide a way of demonstrating that in principle, a hypothesis, a teleological hypothesis in neuroscience, is a valid hypothesis. Even if they can't prove it, because of course you can only prove it via actual data, they allow you to demonstrate that a hypothesis is at its core a very reasonable hypothesis that allows you to make other predictions about what the other purposes of other modules in the brain might be and what an animal's behavior would be based upon certain phenomena. So just as a little primer, so artificial neural networks, of course, most of you are, probably all of you are familiar with, but just a reminder that these are highly abstract models of neural computation. They treat each neuron as a unit, quote unquote, that just sums up its inputs and then runs that summation through some nonlinear function. Now, obviously real neurons are more complicated than that, but at a very high level, that is what neurons do. And so when you string together a set of units that sum up their inputs and then run it through a nonlinear function, you've got something that resembles real neural computation on some level. In part, what you've got is, most importantly, what you have is a system that allows you to study computation in parallel distributed systems. Because that's what the brain is. It's a parallel distributed computer, and so are artificial neural networks. So you have these models which are, at their core, also a parallel distributed computer, just as the brain is. And the thing that's great about these models is that we have good tools within artificial intelligence for optimizing these networks. Indeed, this is why we now associate artificial neural networks with AI. Most people, if you talk about artificial neural networks, will think you're talking about artificial intelligence. Because indeed, like all modern artificial intelligence almost all modern artificial intelligence is done with artificial neural networks. They have totally come to dominate the field because we can optimize them very well for specific tasks. It's worth noting, though, that though we associate these models with AI nowadays, their origins were ultimately as models of the brain. So the earliest such models from people like McGill and Pitts were explicitly about modeling the brain. And even when we look at researchers like Rosenblatt, pictured here with his perceptron research program, who most people associate with artificial intelligence, he was very clear in his writings as well that he considered himself to be modeling the brain. And the modern era of neural networks was, of course, set off by the parallel distributed processing group that, again, were aiming to model the brain. So though artificial neural networks are ultimately associated with AI nowadays, at their core, they are abstract models of the brain. And critically, they are models that we can optimize for specific purposes. And this is why they're good for answering teleological questions. Now, the ingredients that we have for artificial neural networks, both to understand them and to design them, are the following three ingredients. These are the core ingredients that we outlined in a perspective piece in Nature Neuroscience in 2019, which has, I think, helped the field more broadly think about how to describe the ways in which we generate theories in neuroscience using artificial neural networks. So critically, in artificial neural networks, you don't program the network explicitly to do anything. This is, of course, what marks them as distinct from traditional AI models, and also what marks them as distinct from many forms of model used in more traditional computational neuroscience. But instead, with artificial neural networks, what you do is you specify first an architecture. That architecture just means you specify the different groups of units, aka neurons, in your network, and how they are connected to each other at a macroscopic level. You don't specify the fine-grained connectivity, though. You're going to allow your optimization techniques handle that for you. But you do specify a large-scale macroscopic architecture for your neural network, saying this module of units connects to this module of units, but not this module, etc. Once you've defined your architecture, you then have to define an objective function. Your objective function just says essentially what the goals are in your modeling. What are you trying to optimize the network for? So this could, of course, be your error on some task. It could be a measurement of the probability of the data you receive, or I should say the negative log probability. So you want to minimize the negative log probability, therefore maximize the probability of the data, etc. There are numerous objective functions that you could decide on. And then lastly, you need to pick a specific learning rule. So based on the objective that you have, how are you going to change your synaptic strengths in order to either maximize an objective function or minimize a loss function? Here I'm illustrating, I don't actually know if this functions as a pointer. I don't see that. No, okay, I'm not going to do that. So here I'm showing an objective function surface that we're going to try to go up using our learning rule. Now, in artificial intelligence, everyone uses some form of gradient descent because it works really well, and we can discuss later if you want why that is. I personally think that the brain has something like gradient descent going on inside of it. But of course, you're not required to specify a gradient descent-based learning rule. You could specify a different learning rule if you want. But the point here is that if you're a researcher building artificial neural network models, you have to specify these three elements. You have to specify the architecture, the objective function or loss function, and the learning rule that you're going to use. And once you have those three ingredients, you can then optimize your neural network to achieve some task. Now, why would you do that? And how would that help you answer a teleological question? Well, so let's go back to that example I gave of grid cells. So what purpose do grid cells serve? Why do you have grid cells in your brain? I assume everyone here is familiar with grid cells. Of course, the Mosers won the Nobel Prize for having discovered them. Not too far from here, just on the bit further east in Scandinavia. And so, you know, grid cells were obviously fascinating, just seeing these cells that respond to particular spatial locations in a triangular grid or a hexagonal grid, depending on how you view it. The fact that the brain contains these cells is fascinating. But what is their functional purpose? Why would we have evolved to have these cells, or why would we learn in a way that would lead to these cells being produced? I.e., we want to ask that teleological question, why? Why do grid cells exist? Well, in this paper from DeepMind in 2019, what they showed is that in principle, one potential reason for why you could have grid cells would be indeed that they help you engage in vector navigation. So specifically what they did is they built a neural network, which is illustrated on the top left there, where the neural network receives as input an initial position for some agent in an environment, and a set of velocities over time. So the agent is moving through the environment, and the network receives information about the initial position and the velocities of the agent. And then its goal, it's the thing it's optimized for, is to successfully predict where it's going to be based on that set of velocities. So now, where am I going to wind up based upon this changing signal of velocities that I'm receiving and the initial position I was in? And as you can see in the top right, the network learns quite well over time through training to minimize that error. It's very good at saying where it's going to be after taking those various velocities. And lo and behold, what you see emerge in the neural network, in the guts of the neural network there, in the LSTM, is, or maybe it was in the linear layer, I'd have to go back and look at that. But what you see emerge in the guts of the neural network, as shown here in the bottom rows, is grid cells. And furthermore, they showed in that paper that not only does optimizing for this vector navigation task lead to the emergence of grid cells, but then you can demonstrate that the network has all sorts of other abilities related to vector navigation, and if you lesion these cells, they can't do it, et cetera, et cetera. So it allows us to perform the sorts of experiments that we couldn't perform in animals using these networks that we're capable of optimizing quite well. Here's another one that I thought might be interesting for this crowd. I know many people are interested in the question of hyperconnectivity in various neurological disorders. For example, this isn't my area of expertise, so I apologize if I'm about to say anything wrong. My understanding is that autism contains, autism is often correlated with hyperconnectivity in the brain. So why would that be bad? I mean, why would having more synaptic connections harm you? At face value, it actually seems to me like it should never harm you. If I add more synapses to your brain, you should just be better at things. It shouldn't impair you. It certainly shouldn't make you handicapped. But in this paper from Raman et al., from Kevin O'Leary's lab at Cambridge, Timothy O'Leary's lab at Cambridge, what they showed was if they take a neural network and they train it on some task, so they just pick a fixed mapping and they train the neural network to recapitulate that mapping, whatever it is, by minimizing an error. And then they add some noise into the neural network's learning rules. So now it's doing gradient descent plus some noise. What they find, interestingly, so they actually derive this mathematically, which, for the record, is, again, an example of what you can do with artificial neural networks that you can't do with other models. They derive analytically a demonstration that if you have some additional synaptic noise added to your neural network, then at some point, a network could actually have too many synaptic connections. They also show that empirically, which is what I'm showing here. So here you're looking on the left on these two plots at the training curves for neural networks of different sizes. So the neural networks are all learning the task to some extent, but they're plateauing out at different levels of error. And then on the right, you're seeing the error that they plateau at as a function of the number of synapses in the network. And if you don't have noise in the network, which is what you see on the top row, there is no maximum size that's best. It matches what my intuition was. More synapses is always better. You will always do better on the task the more synapses you add. But if you add some noise to the synapses, now you get a diminishing returns. And in fact, as you continue to increase network size, the network will get worse and worse. There is, in fact, a specific size of network that is optimal for the task. So again, this answers the question, why would there be an optimal number of synapses? Why isn't it just the most synapses possible? And this neural network model allowed the authors to demonstrate that one of the potential answers to that question is because if your synaptic update rule has some noise to it, then necessarily there is such a thing as being too big because the noise from all those extra synapses starts to swamp your learning. As a final example, I wanted to show you one that was a Highball and BigBrain funded, or sorry, I should say, Yeah, a highball and HBHL funded project. This was a project from my lab led by a former postdoc of mine, Shahab Bakhtiari, who's now a professor at Université de Montréal, an assistant professor at Université de Montréal. And we asked the question, why does the brain have segregated visual pathways for what and where? So why do we have a separate dorsal and ventral pathway that seems to process movement versus static features respectively? So what we did is we took a neural network model shown on the left here, and we trained it to predict upcoming images. So we trained it on movies. So the neural network received a movie, and it had to predict the next frames of the movie based on the most recent frames. And then what we did is we looked at the similarity in the neural network to different parts of the mouse brain. Here we're showing 5 different regions of the mouse brain, VisLM, VisP, VisPM, VisAL, and VisAM, where I've arranged them from being more ventral to dorsal. So the more ventral ones, they're the what pathway, like VisLM, and the more dorsal ones, they're like VisAM, they respond to movement in the mouse brain. And what we found is that when we built a single neural network pathway and we trained it on this visual prediction task, it would develop something that looked like the ventral pathway. This can be seen from these plots because these are plotting the similarity, the representational similarity of the neural networks to those different parts of the brain across the layers of the neural network. And whereas you can see that in the more ventral areas, there's an increasing similarity to the ventral parts of to the brain in the neural network, in the more dorsal areas, there's a decreasing similarity. So when we trained this individual neural network, we saw just a match to the ventral or what parts of the animal's brain. But when we added 2 separate pathways, so we altered that architecture that I was talking about, the macroscopic architecture of the network, then what we saw was actually that one pathway would come to look a lot like the ventral parts of the animal's brain, and the other pathway would come to look like the dorsal parts. And we were further able to show that that helped the network with the prediction task. So, in other words, what is the purpose of segregated pathways? It actually seems to help a network engage in visual prediction. And we argued that the reason is because when you're trying to predict what you're going to see in the future, you are aided by being able to predict both what you're going to see and where different things are going to be moving to. So naturally, this is a functional division that emerges from optimizing a network with these separate anatomical pathways. So to wrap up, my central point here is that artificial neural networks provide a means of asking teleological questions courtesy of their trainability. In some ideal world, we would be doing everything in highly biologically realistic models that capture all the details about synaptic receptors and dendrite shape and ion channel ion channel densities and all that kind of fun stuff. But the problem is that biological models like that, biophysical models, are completely handicapped in terms of our ability to optimize them. You can't train them to learn any useful tasks, in part because they're too computationally expensive, but also because they're not mathematically tractable for optimization in theory. Moreover, they have way too many hyperparameters to control, and you can't take advantage of all the tricks that all those AI engineers are constantly cooking up to get better at optimizing the thing. So artificial neural networks are really great for this. To put it in another way, our ANNs are in a Goldilocks zone, where they have just enough similarity to the brain such that we see related phenomena emerge in them because they too are parallel distributed processing systems. But they are abstract enough to make learning possible. And teleological studies where you ask the why question can only be answered when you have a model that can be optimized, that is sufficiently tractable to be optimized. So they sit in this Goldilocks zone that allow us to ask these why questions. As a note, a final closing remark, we have developed a tool to make it easier for you, for anyone, to build artificial neural network models based on the anatomy of the brain. So that architecture component that I mentioned, ideally the architectures would actually match the architectures of the real brain. And various data, including data from the big brain, could help us answer teleological questions with greater correspondence to the real brain if we had ANNs whose architectures matched that of the real brain. So we've built a tool to allow you to do just that, to use a combination of tractography and big brain histology data to build ANNs based on the macroscopic architecture of the brain. I'm not going to tell you any more about that, though, because my student, Mashbayar Tugsbayar, is here, and she's going to be talking about that in contributed talks too, and also has a poster on it. So little advertisement there if you're interested in building your own models based on big brain data. That's that. Thanks very much for listening.
All right, thanks, Blake, for the great talk. What I'm going to do is to pick it up where Blake left off with the teleology talk. So I'm going to go beyond that. And hopefully I'm going to tell you a bit about another perspective, another direction, another approach that you could use these sort of neural network models in a way that I and maybe some others consider more practical and more applied. So let me just get started by going over maybe a brief explanation of the setup that we have in building these neural network models of the brain. So we have two systems. We have the brain as our biological system, and we have these neural network models that Blake was telling us about. Within the framework of deep learning, we essentially abstract them as a few knobs, a few control knobs that mainly constitute architectural parameters, learning rules, and learning objectives or cost functions. So three things go into these neural networks, and we build these neural networks, and what happens is that then we expose them in parallel into various sensory experiences. And these sensory experiences could constitute things like auditory signals, tactile signals, or visual signals. So these things go in parallel into these systems, in vivo and in silico. And each of these systems generate outputs. And most people that are active in this domain of computational neuroscience, what they do is essentially they observe the output from these two systems that are generally, you could consider them as what's going on inside in terms of like internal activity, which in the brain we consider neural activity, for example. And in these neural network models, we consider them as activations, unit activations, and also the output that they generate, which would be behavior in terms of animals, and the actions that these neural network models generate. And to sum the whole thing up, what people do is essentially try to look at how similar these things are. How consistent are the behaviors? How consistent are the internal activations? And going after those why questions, essentially people take different hypotheses in terms of what best explains what's happening in the brain or the behavior that the brain generates, in order to kind of vote for competing hypotheses within the teleological questions or why questions. So as I said, I'm going to go after what I consider a more applied direction in using these neural networks. And I originally had the title as How to Build a Brain Model, and then I had a discussion with Blake, and he somewhat convinced me that those other models that do not necessarily correspond to units in the brain, you should still consider them as brain models. So I added this parentheses, how to build an actionable brain model, and I'm going to tell you why I consider these actionable in contrast to the type of models that Blake was telling you about. So there are two ways you can make actionable models of brain. One is that you take your neural network and imagine what you're after is to really explain what a neuron or a voxel in the brain, the activity within a voxel in the brain, is doing. What is the function that describes what this model, what this neuron is doing, right? One way to do this is to go inside your neural network model, look at every unit, and try to find the unit that best matches with what you're observing in the brain, given that it's a neuron or a voxel or whatever. This approach essentially assumes that your units in the neural network model are aligned. You would find some aligned units in your neural network that exactly match what's going on within a neuron or a voxel in the brain, which is a very hard constraint. But what people actually do in practice is to go by a many-to-one mapping, which, long story short, is that instead of considering a single unit, people usually go inside and consider a combination, a weighted combination of all or some of the units in the neural network model to be producing an explanation or a function of what a neuron or a voxel is doing. And this is kind of relaxing the assumptions by considering that you could combine these neural network units in order to align them with what's going on in the brain. And in practice, you usually get better answers, better solutions, better explanations of what's going on also. So they're practical. Now, let's say we have the brain model, but what can we really do with the brain model? So that's what I'm going to tell you next. One of the main things that people do with these models is to predict. And prediction is a really driving force of many labs working in this area. And the kind of questions that they try to answer is things like, such as, how would neurons respond to new stimuli? Like stimuli that you haven't seen when you were running an experiment. In the case of visual neuroscience, you could consider showing static images to animals and then recording brain activity from them as they were observing them, and then make a predictive model or make a model of how a single neuron is responding to these static images. And then what you actually want from your model is that it would be able to tell you how would this neuron respond if tomorrow I will be showing it a bunch of new images that I don't know how it would respond to, right? So it's kind of like make the job of the experimenter easier so that it wouldn't have to collect tons and tons of data to explain or tell people how a single neuron or a single voxel would respond to all possible stimuli in the world, right? And another kind of questions that people go after is to try to answer how would neurons respond when performing a new behavior. So it's kind of like generalizing the stimulus into domains of behaviors, like what if the animal is actually doing a task which is dependent on visual stimulus versus just like fixating? Or what if the animal is trying to memorize information? How would that affect the function of that neuron? So you essentially want a model that would give you correct answers under all of these different circumstances. And finally, another direction that I think these models would be really helpful is to help us, or maybe physicians, to try to predict how neurons or the brain or behavior that the brain generates would be affected by a lesion or by an injury, for example. So these are kind of the broad questions or example of broad questions that you could go after with these sort of neural network models. And some of the classic examples of this work now dates back to almost a decade ago, which was actually, this work was my great inspiration of doing my first postdoc in Jim DeCarlo's lab, which what they did was that they used a particular architecture of these neural networks called convolutional neural networks to do object recognition, and then they showed that when you looked inside these neural networks, you find that combination of unit activities, they predict really accurately a lot of the neurons that you find in high-level visual cortex. And at the time that they published this work, people in the field really were scratching their heads trying to figure out how is it that neurons in IT cortex respond to such complex patterns, and how is this pattern generated? And this model was really giving the first level solution or first level computational explanation of how is it possible to get such a function out. And that was in the visual cortex, visual domain in vision, but the later work was not limited to that domain. Other groups started to look at other sensory cortices, in this case into auditory cortex, and they developed somewhat similar to a class of neural network models, again here convolutional neural networks. At the time, and still, they were very popular because they're very efficient and they work really well. And they were able to explain neural activity across different parts of the auditory cortex. And another nice thing about it, which I did not mention in the previous work, was that when you look at the layers in these neural networks, you see this relatively good match between what we know about the hierarchy of information processing in these sensory cortices and the layers of these neural networks that you have. It's like, let's say you have 3 layers of neural networks. The first layer usually corresponds better to early stages of processing, and the later layers best correspond to later stages of processing in the brain. And I told you mostly examples from modeling sensory cortices, but now what we see in the literature now has gone much beyond sensory cortices. We have a lot of examples from parts of brain in across species that are related to memory, decision-making, language, motor, and you name it. So the field has really expanded since a decade ago. And you might be asking, maybe prediction is not really enough. Some people would actually say that, because they think just predicting and predicting what the response is gonna be is not necessarily give us any sort of understanding of what is really that neuron or voxel in the brain area is doing in a way that we as humans would understand it. So just to make a case, prediction on itself may not be sufficient for gaining understanding, but it would be a core part of a tool that could lead us towards a more precise and computational forms of understanding in some ways that I'm going to just mention 2. One is to reverse engineer. What I mean by that is that, say you have a neuron from some brain area or brain area activity that you're really interested in knowing what function it serves, right? And without a neural network model, what you need to do is to keep experimenting, keep probing that area, and keep looking in the space of possible hypotheses that you, as a human being, as a scientist, could come up with in terms of the function of that brain area. But now having these neural network models, if you don't really have a great idea of what that brain area is doing, but you have a good predictive model, you could go and look inside your predictive model and try to put things together in terms of connectivity between units and what each unit in that neural network does to form somewhat like a description of how such a function is generated by a combination of different lower level features that may exist in lower layers, lower brain areas. And another direction that you could go is what I'm calling control here, and I will be talking a bit more in the next slides, and that is I consider this as a really good tool, a really valuable tool in terms of discovery. And what it means is that you could now use this sort of neural network model and then hypothesize about any sort of brain responses in an area or across areas, and then this model would enable you to go back and discover what sort of experiences would lead to that sort of responses in the brain. So it's kind of like going backwards from output, imagining a possible output in the system, and then going back to the causes in the experiences in the sensory area that would lead to such responses in the brain. And I'm going to expand on that a bit more. Before I get to examples of this, I put this here just to give you an illustration of what I mean really by the process that I'm calling control. Let's imagine you have this. You see my mouse pointer? Yes. So let's imagine you have a predictive model of neurons in one brain area, let's call V4. What you could do is that when you construct this mapping that goes from image pixels to neural activity in every neuron in that area, you could now use this neural network model in reverse mode and go back through the model and find pixel patterns that would generate a particular desired response across the population of V4 neurons. So you could design, for example, what is the pattern that really excites one neuron and not any of the other neurons in V4 area, and that would give you a sense of the selectivity of single units in that area. This is just an example, then I'm going to talk a bit more about it. So people actually tried this procedure in 2 different brain areas, 2 different species. There was work from Andres Toius's lab looking at mouses V1 and making a predictive model of neurons in this area, and then using the model to try to construct what they called the most exciting images, which really excited those, made the neurons in area V1 to fire really high. And using this model, they showed that the previous ideas, the previous beliefs about the selectivity of V1 neurons in the mouse's visual cortex was not really an accurate description of what these neurons in practice do. Because previously people believed that these neurons are most selective to gradings of different orientations and spatial frequencies, but their results showed that the response, the selectivity of these neurons is much more complex than just that. It contains corners, it contains even some sort of curvatures in mouse V1. And another work which came from Jim DeCarlo's lab, and I was also part of it, was kind of applying this procedure in another area called area V4 in primates, and kind of showing that, again, in this area, the classic beliefs of V4 neurons responding to curvatures of convex and concave halls, they're not really accurate descriptions. The parameterized space is not capturing everything that these neurons are doing. And the real answer seems to be a combination of different textures, different shapes that may have many more parameters than just like being concave and convex with few parameters. And you could, so far I talked about single neurons, trying to discover what single neurons are doing, but you could apply the same procedure to population of neurons. You could try to figure out what the population is really selected for, like what are the different axes that the population of neurons are coding. And you could go individually, select single neurons in a population, and try to activate one by one without affecting any of the other neurons. And again, get visual descriptions of what population code along a single axis would mean. As a final example, this sort of direction has been expanded to other brain areas. In this case, it was extended to a brain area that was underlying language comprehension. And they used a similar method in making predictive models of voxels in that area. And then using the model to select, now this time, select what they considered as the most exciting sentences and least exciting sentences. And then they tested it in practice using experiments, and they showed that what the model considered or found as the most exciting and least exciting sentences were in practice driving that language area most and least. Now, I talked to you about a bunch of different examples and a more applied direction that you could use these neural networks. But what do we really need to succeed? Of course, on one direction, we need just more capable neural nets, like bigger neural nets that could generalize better in terms of stimuli, in terms of different tasks. We need better models, better neural network models that are more robust, that their predictions are not highly affected as they are right now by patterns of noise, for example. And we know that in the human brain, a lot of the decisions, a lot of the perceptions that are generated in our brain, they're much more robust to many forms of perturbations to the inputs that we receive, to the sensory input. And we currently lack a lot of that in neural network models. But that's not the whole story. From the other's perspective, we really need more detailed brain maps at different scales. An example of that would be more detailed anatomy and connectivity, such as those that we have seen today, and hopefully we will be seeing more of that tomorrow and the day after from the BigBrain project and the eBrains project. And also, we would need larger data sets with more samples and tasks that are functional data sets that would tell us how the brain activity changes across different stimuli, different tasks. And examples of these are, maybe you're familiar with some of them, the natural scenes data set, in which a few subjects, 8 subjects, data was collected from their visual cortex for about 10,000 images within each subject, and overall around 90,000 samples, which was kind of multiple orders of magnitude more than any other data set that was out there, and is really extremely helpful for the whole field. And the Neuromod project, being led by Dr. Pierre Bellec at the University of Montreal, who are, again, following a similar approach, taking few subjects, 6 subjects in this case, and asking them to do a broad range of behaviors, including playing games, viewing episodes of Friends, viewing static images, and performing working memory and different sort of memory tasks. So with that, I'll just sum up. It's open to questions, both from me and Blake. I don't know how much I went over time, but hopefully that's not too much. Thank you. Go ahead. You can go ahead.
Here, you can take this.
I'll do the running around again. Hi. So I have a question. So neural networks are known to be high complex models. So how can this low interpretability be handled for the reverse engineering process? So because some authors even categorize neural networks as black box models. So that's my question. Thanks.
I imagine that's directed to me. Yes, so I'm going to give you one answer. Maybe Blake wants to give his own version. I think the argument of saying these models are black box models and we cannot really interpret what's going on in them is not really a valid point anymore, because we have so many tools for visualization and for interpreting what is going on in the units of these neural networks. There are tools, for example, in the vision domain that you could target a particular unit in a neural network and search within static images or video to see what features they're tracking. Like the sort of techniques that I showed you in terms of applying control are some of those. You could try to find out what are the optimal stimuli or the conditions that lead to activation of a particular unit or particular ensemble of units in these neural networks, and give description in terms of what sort of experiences would lead to activation of that unit. So I agree that maybe a decade ago we were handicapped in that domain, but the field has moved on, I think, and now we have many tools for doing those sort of things. It's just a matter of like, what would be the level that users are interested in interpreting their models. And now there are tools for various levels. Maybe they're not all perfect, so that you cannot go like, you may not be able to answer all of your questions, but a lot of them could be answered. Do you want to add something?
No, not majorly, but I think I'll just add that the other thing is, because, as I said, these models are mathematically tractable, you can still also engage in a sort of reverse engineering via the geometry and other mathematical analyses of these networks. It's just that maybe it's not always easy, though as Pouya is describing, this is not necessarily the case anymore, but it's not always easy to say specifically what each unit is doing. But there's a part of me that's also just comfortable with giving up on that way of describing how neurons work, period. And I personally have argued that that's something that neuroscientists should give up on altogether.
It's probably too extreme.
Yeah.
So I was told to be provocative. So it's fascinating stuff, and I'm sure you've thought about these problems a lot. But everything here that you're describing, I think, is architecture dependent.
Yes.
Right? So you can optimize the models to get the same performance as a human brain, but you have no idea if that is modeling the human brain. It's just producing equivalent results. So what do you do with the temporal component? We know that all these regions are connected to each other. We know the what and the where in a different architecture, but what about the when? This node is temporally very close to that node. This node is very far from another node because of the signal propagation time between those nodes. To what extent is that information taken into account? All nodes are not equivalent. All spokes are not equivalent. Every one of those is different by definition, by evolutionary standards. So how does that information get brought into the equation? Now, Blake, you said, yeah, we can hyperparameterize and it'll be insoluble.
Yes.
Yeah, well, that's that may be true, but it's also how the brain actually works.
Right.
So this is the issue. How can we model the brain if we don't include the brain's real-world properties?
Do you mind if I start with a response to that?
I'm sure you've had that question many times.
Yeah, so yes, there's two answers to that. The first is, of course, if you're really concerned about the delay times and the temporal order of activations, you can develop models, and people have done this, where you introduce different time constants on different connections within the network. Now, of course, as you noted, that means you're introducing another hyperparameter that you don't have to control, but as long as you do a minimal amount of that, it's still optimizable. But I think it's worth noting as well that there is some implicit delay built into some of these models insofar as if you have two modules that are connected via two synaptic jumps versus another set of modules that are connected via one synaptic jump, that two synaptic jumps will take literally twice the number of computational steps for the model, for module 1 to affect module 2 versus the one synaptic jump connection. So when it comes to simply the number of synapses, we actually already kind of capture that delay to some extent. What we don't capture is literally the length of the connections. So say that it's a much longer connection from prefrontal cortex to primary visual cortex than it is from prefrontal cortex to motor cortex. But I think these are the sorts of things that in working towards brain models, eventually, indeed, we will want to incorporate. And the key is, in my opinion, taking baby steps where you always introduce just additional components of detail without breaking your ability to optimize the model. Because for me, that's the key problem. Once you've gotten to the point where you can't optimize your model, now it's no longer useful for answering teleological questions, for example, and probably less useful for prediction and control as well.
Yeah. Maybe I'll just add one comment on that. We maybe have been focusing too much on processing static inputs into what we presented, but that's not by any means the whole field. Yeah. There are these other classes of models called recurrent neural networks that are just designed to process sequences of inputs. And you could design them in a way to somewhat implement some aspects of what you're touching on. For example, we have different brain areas. If you look at the spiking activity, you see this like progress of spiking activity going from one area to another area, coming back to another area. So all of these, in theory, are implementable in even these neural network models that are really abstract from the reality of what's going on in the brain. But if we agree with Blake, and maybe me, that these ANNs are at the Goldilocks zone of current technology, it may not be actually, these networks may not be the Goldilocks zone of technology a decade from now. Yep. In a decade, our GPUs and our hardwares may have progressed so much, our software have become so much more efficient that Blake would change the slides and putting biophysical models into this and then saying that Goldilocks zone is now ANNs with biophysical properties, but not ion channels. Right. So it's like this is an evolving field, and current models, they have some capability in that regard. There are clear indications how they could be improved more, like incorporating biophysical properties, delay parameters, and so on. But I don't believe that would be the end of it either. We probably will get to a stage at some time in the future where we will be capable of incorporating even more about what we know about the brain. It's just like people now want to do something that is interesting to help them discover something about the brain that is doable at the current time. And that's why a lot of what we see, people try to abstract away, because you always want to do something that is doable. Yeah.
The second point that I thought would be interesting to raise is Blake, you made the point about that if all things were possible, everything would be connected to everything else. That would be the fastest. And there's work, I'm sure you're familiar with the work of Bullmore and Sporns, the economic energy minimization constraint, which is, that costs. So you can't have everything wired to everything else. Otherwise, evolutionary speaking, that would die out. So you come up with small world models where you have long-range connections between different parts of brain, which reduce the amount of connections you need to achieve the same output. So there's a natural constraint pushing back at the other end. So there is an optimal architecture, which involves small world connectivity. Of course. So can you build those into your models?
Oh, certainly. So you can definitely build small world connectivity into these models, and you can explicitly try to mimic the macroscopic architecture of the graphs that we see in the brain, quote unquote, with the graphs that you build into your neural network. And I think building in additional considerations like energy constraints and stuff like that is another interesting why question, and it's another way that you can potentially answer this. So with the example that I gave of hyperconnectivity, I was ignoring the question of like energy and distance constraints when I said my intuition would be that you'd want as many synapses as possible. Because what I was speaking of there is just from a computational perspective. If I'm just trying to compute a large number of functions, I want as many synapses as possible. But you're right that when we start to add these other considerations, there can be additional why questions beyond those computational ones, such as it helps with you know, the amount of energy required to wire up your brain in early life. But I think it's also then interesting with respect to these individuals with hyperconnectivity, given that you know, we live in a society where evolution's not selecting against them as strongly, so they get the chance to survive. Again, my intuition would have been that they would be superior cognitively to others, and they're not. And that's interesting. Questions?
Thanks, first of all. I think it was an extraordinary lecture, also in a cool partnership, so it was really nice. My small question would be just if you think about that we sometimes wire new connections, right? So the architectures you described are mostly in our tool sets very fixed. So we cannot just jump, you know, a couple of layers like in ResNets or something. So did you have thought about these approaches? How you model this kind of new wiring up, especially if you think about longer periods?
Have you thought about this? Because I haven't answered.
Sorry, I wasn't really clear on the question. Can you sum up your question again?
No, sure. When I think there's a concrete CNN that you have, right? You have a concrete architecture, how many, you know, layers, how many weights, and so on. But when you think about humans, how they learn, or maybe even unlearn, interesting question for why as well, why we unlearn and have sometimes not spikes, but losing maybe connections. I mean, something where the architecture is not fixed, but changes during the time in order to learn a task better, in order to forget. Maybe.
Ongoing plasticity.
Yeah,
So
okay. I'm not the neuroscience specialist here.
Well, I think also, yeah, like the pruning of connections or the addition of new connections is what you're getting at. An architecture that changes over time rather than an architecture that's fixed from the get-go.
Going beyond regularization, it's more.
Yeah, yeah. I think that this is an area of active research in ANN research, in part because there's a push from within the artificial intelligence community to develop techniques for pruning away your neural networks so that they're less compute expensive when you actually deploy them. And there's a whole fascinating area of research based on something called the lottery ticket hypothesis about why you want to start with lots of connections, as you see in the brain, and then potentially prune things away over time, over development. I won't go into it here because it would be a whole other lecture, but for those interested, if you're interested in these questions of like changing connectivity over time, and in particular the tendency for the brain to remove connections over time, I strongly recommend checking out this idea called the lottery ticket hypothesis, which provides a mathematical explanation for why that might be a doable thing in the brain and helpful. Thank you. Any questions? Looks like we've got more up there.
Okay, great talk. As an example, if we just think about grid cells, so most mammals have grid cells, and I'm just wondering then, do you think that this is something that's biologically coded, or do you think that this is something that is generally emergent? So in other words, are something like grid cells, and I think this applies to your other examples as well, are they genetically or biologically encoded, or are they a universal property of space?
Right. So the answer to that at the current time, based on the current available evidence, is an interesting one. When people look in baby animals, they don't see grid cells per se right away, and in fact, grid cells emerge after place cells. However, what they do see right away is that activity in entorhinal cortex is restricted to a toroidal geometric structure, basically a ring in high-dimensional space. And that ring in high-dimensional space is part of the structure that allows grid cells to function in the way that they do. So what's interesting is it looks like there are some genetically encoded priors in the architecture that are kind of ideally situated for representing space with grid cells. And then during learning in life, actual grid cells emerge from that.
Cool, thanks a lot.
Last question there. We could go on for hours, but we have
Yeah, sorry we ran right up to the butt of time.
First of all, from me also, I loved the talk. I have three, but I'm just going to ask one. And I have one comment for what you said about the time component, the temporal component. To my knowledge, there is one paper from 2016, if I pronounce the name correctly, it's like Kichi, something like this. So they show that there is an emergence of a temporal component, even though it's not encountered in that model. So they do it by having MEG recorded while they show the images and correlate it with each layer of the networks. And it shows very well that there's an emergence of this temporal component of processing inside. So even though it's not considered in the modeling, it's inherently somehow in the model. But on the other hand, I wanted to ask, so neural networks are inspired by the brain for like, they became what they are from just our small understanding what the neuron does. So they are kind of like two neuroscience in parallel. One, it's neuroscience with pure computer science, and the other is neuroscience with science. To which extent can this go and break after a point of our understanding? Because, for example, neural networks is just, let's say, a high-level description of what computational neuroscience is trying to do to understand to the molecular level of what the neuron does. And how does this limit our architectural understanding of the thing? So that was the question.
Do you want to start with your response?
Sure. It is already limiting. Examples of that would be when you have these neural networks of that model neurons, let's say in prefrontal cortex, because these units are not being distinguished based on different receptors that they have, we are unable to give any sort of explanation of what would happen if you inject a particular neurotransmitter, for example, dopamine or say will, right? So they're already handicapped in terms of the questions that could be answered using them. But right now, people are really focused on questions that concern representation. How is it that the solution emerges? And then kind of like putting a lot of the details under the rug just to be able to provide a first level answer to how solutions to some of these behaviors might be emergent across different brain areas and in combination leading to the behavior. But yeah, I think that would be my answer, that they're already handicapped. It really depends on the level of question that you're asking.
Yeah, I will largely just second that. I think that, you know, obviously I was kind of giving a pitch for why you'd want to use these models, but I don't mean to imply that they are the tool for answering every type of question. So per what Pouya was just saying, there are many questions, e.g., for example, trying to answer questions about, you know, different types of receptors or something like that, that the current models are not going to help you answer. I think, and this gets back to some extent what I said to Alan, and kind of what Pouya was pushing at as well, with relation to the question of what is the current Goldilocks zone. The hope is, the dream is that we will be able to eventually patch these divisions that come up and that prevent us from answering certain questions, either as a result of increased computational capability or new tools that allow us to optimize well, even with the presence of these additional components added in. And I think we've already seen some of that in neural networks research. We've seen that we can include, you know, much greater recurrence. In my lab, we've shown that you can include different types of recurrence with different types of top-down feedback. It still works well. You can include dendrites, and it still works well. There are certain things that you can do. So the hope is that this is to use the phrase from Tim Keitzman in his paper that he led and I was a co-author on neuroconnectionism as a research program. The hope is that this is a productive research program, that we continue to generate new ways of doing things and new insights because it's a fundamentally productive paradigm for operating within, a language for doing science within. But that's not to pretend that there aren't clear divisions that have already emerged from these two parallel ways of thinking about things.
Can I just add one comment on what Blake said? In a way, this neuroconnectionism research program, you could disagree with me, is in a way infalsifiable. Because if you really think deeply about what neural networks are saying, if you believe that the brain is computing, and you would be able to approximate or write down the function that the brain is computing mathematically, then essentially you cannot escape the fact that some version of neural network models that underneath it all is just like mathematical equations, mathematical computations, cannot explain. So it all goes back to believing whether brain is computing and that's it or not.
I will just say, I think that's not wrong. It's kind of like saying, you know, do you want to do physics without calculus? Like, no, it's a good language for doing physics with it.